Meet Marcel, the cell. Marcel enjoys long walks on the beach and meeting different egg cells. That's right, Marcel is a sperm cell. But where did Marcel come from? Like you and me, Marcel came from his parents. Well, parent cell, that is. Marcel came into the world as a result of either mitosis or meiosis. Both are methods of cell reproduction. Mitosis produces two daughter cells from a parent cell that are identical to each other as well as the parent. Meanwhile, meiosis produces 4 haploid cells from one parent cell. So which method of cell reproduction gave us Marcel? That depends on if he is a haploid cell or a diploid cell. So what exactly is the difference between a diploid and haploid cell? Diploid and haploid cells differ by the number of chromosomes present in the cell. Let's say that N represents the amount of genetic material present in a cell. In humans, these are chromosome pairs. Diploid cells have 2 N chromosomes and haploid cells have N chromosomes. Therefore, if Marcel is a human cell, his genetic material would consist of 23 chromosome pairs. If he is a diploid cell, he would have a total of 46 chromosomes, or 2 times 23. If he is a haploid cell, he would have a total of 23 chromosomes, or 1 times 23. The first thing Marcel goes through is mitosis. Mitosis starts with 2 N chromosomes and 4 times DNA content, because each of Marcel's chromosomes has replicated. These chromosomes condense and line up at the middle of the cell. In anaphase, the identical sister chromatids separate and go towards opposite ends of the cell, where they eventually pinch off into 2 daughter cells. These daughter cells are identical to the original cell, each with 2 N chromosomes and 2 times DNA content. All eukaryotic cells go through mitosis, except for cells that create gametes, which are eggs and sperm. They go through meiosis. Meiosis starts off with 2 diploid cells, each with 2 N chromosomes, and ends with 4 haploid cells, each with N chromosomes. Meiosis involves two divisions, meiosis 1 and meiosis 2. Meiosis 1 begins with homologous chromosomes lining up next to each other, and an event unique to meiosis occurs, crossing over. 2 homologous chromosomes exchange DNA between adjacent homologous chromatids. Now, the sister chromatids of one chromosome are no longer identical to one another. At this point, each chromosome is still double-stranded and has 2 sister chromatids. Therefore, in meiosis 1, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After meiosis 1, meiosis 2 starts a second division, which is identical to mitosis. At the end of meiosis 2, we see the formation of 4 genetically different haploid cells. Because Marcel has a total of 23 chromosomes, he is a human haploid cell created by meiosis. He has 3 siblings that also have 23 chromosomes. Marcel and his siblings don't have the same exact chromosome makeup because meiosis doesn't create exact replicas of the parental cell. During reproduction, Marcel and his siblings' different chromosomes become apparent. While human siblings do look alike and share many characteristics, because the haploid cells have different chromosomal makeups and underwent crossing over during meiosis, each sibling will have different allele combinations. In short, mitosis creates 2 diploid cells that are identical to each other in the parent cell, and meiosis creates 4 haploid cells that have half as many chromosomes as the parent cell and are each made up of unique chromosomes.